My name is Pia Voelker. I work for the German organization called Gen-ethisches Netzwerk in Berlin. We do critical science communication for the interested public in our magazine and in our podcast, and inform, among other topics, about developments in the field of genetic engineering in agriculture. With regard to new genome editing, such as CRISPR, we see a number of risks that make a strict regulation of the techniques absolutely necessary. Some of them are specific risks associated with new genome editing. One example is the application of the technique itself that can cause unintended on- and off-target effects, which are specific to the technical process. For example, there might be an unintended insertion of DNA fragments into the target sequence of the target organism, or there might be effects outside of the desired target sequence. I would like to point out that the use of biotechnological mutagenes enables the circumvention of natural mechanisms of gene regulation and genome organization, and they also make the genome available for changes to a much greater extent than ever before. Genetic conditions, which could be hardly altered by conventional breeding, can now be genetically engineered. But due to the technical processes used in genome editing, the resulting organisms and the associated risks can be substantially different compared to those derived from conventional plant breeding or what can be found in nature. But also the intended effects arising from the application of the technique very often result in specific patterns of genetic alteration and new combinations of genetic information. Typically, this outcome is different compared to conventional breeding, even in cases where no additional genes are inserted. For example, genome-edited plants may change their composition, which can alter their susceptibility to diseases and pests, their nutritional value, and ecological functions. The resulting biological characteristics may be linked to substantial risks for humans, the environment, and nature. The risks are still inefficiently researched, and further research is needed in this area. But beyond the specific risks, there are other problematic aspects associated with new genome editing that make a strict regulation absolutely necessary. It is also about ensuring freedom of choice and transparency for consumers, farmers, beekeepers, breeders, seed production, food companies, and trade, and maintaining labeling, traceability, and liability for GM products. The framework that is needed here therefore continues to include approval procedures with comprehensive risk assessment. In accordance with the precautionary principle, the technology-related risks must be assessed and the labeling and traceability of GMO products must be ensured. Policymakers can ensure this by continuing to maintain the strict legislation in the EU, as the European Court of Justice has already ruled in 2018. Precisely because the very high potential of technical intervention, the risks are often complex. The possible damage often only becomes apparent after a long period of time. Therefore, the precautionary principle must be strengthened.